Night at my by my watch, so we can start. Please start live streaming. Okay, I think it is started. Okay, good morning, everyone. On behalf of Center for Advanced Studies, I welcome everybody this morning in week 4, session 2 of our weekly webinar series, which we organize every week. I would like to welcome the speaker for today, Dr. Pooja Saxena, who has joined us from Canada. Our guest panelist, Dr. Priya Srivastav from St. Xavier's College, Ranchi. Director Sir, Professor Manish Kaur, all the panelists from Center for Advanced Studies, and all the attendees who have joined today on Zoom and who are watching live on YouTube and other platforms. Let us get started. I request Saurabh sir to kindly introduce the speaker, and then we can start.
Okay, thank you, Dr. Vrinda. So, very good morning to all of you. I, Dr. Saurabh, on behalf of Center for Advanced Studies, again, I welcome you all. And let me brief profile of Dr. Pooja Saxena. So, Dr. Pooja is an international virologist with over a decade of research from renowned labs around United Kingdom, United States of America, and Canada, with a strong biochemistry background. So she has built a wealth of knowledge in field of virology and nanotechnology. So during her doctorate studies at the John Center in United Kingdom, she mastered several technologies that are used to develop vaccines and therapies. Currently, she works as a manager and product discovery at multinational pharmaceutical company situated in Quebec, Canada. So her commitment toward the advancement of medical science and development of vaccine for a healthy world is evident from her publications record and great speaker appearances. So she's a BSc in honors in biotechnology from University of Sussex, United Kingdom, and she's a doctorate in virology and in nanotechnology center from John Innes Center in United Kingdom. She completed it in 2013, and she worked as a doctoral researcher in structural she worked on structural virology at Indiana University, United States of America, from 2014 to 2016, and she also worked as a scientist and a researcher in Medicago Research and AMP Dealings, Canada from 2016 to 2020, and currently she's working as a manager in product discovery, Medicago Research and Development Corporation, Canada. So thank you, ma'am, for joining us. It's over to you, please.
It's a pleasure. Thank you for the introduction. I'm just going to start sharing my screen. It's a real pleasure to be involved and to be able to inspire some young minds this way. So I know that our audience is very diverse with people from a lot of engineering, computer science backgrounds as well. So we'll try to talk through some work that I have been pursuing over the last 10 years or so. And I hope you learned something new today from my talk. So really, every time I spoke previously to an audience from an engineering college, I used to spend the first few minutes saying that we're talking about viruses, biological ones, not computer viruses. But today, I think I don't need to spend much time because of the corona pandemic, we know the word virus far too well. It's all over our news feed. Everything we do is influenced by the pandemic that's around us. And it's a sad reality, but it's a reality, and we have to accept that we have challenges like viruses in front of us today, and we might have challenges like this in the future. So it's very important to understand viruses and realize what they are, maybe even learn from them, because at the end of the day, these are gifts of nature. Nature has evolved viruses for several reasons, and we might learn a thing or two that we can apply in nanotechnology, in engineering, from these little nanomachines called viruses. So one thing to realize is that we've been living with viruses for a very long time. They're all around us. They're abundant. If we take just 1 milliliter of seawater, so just 1 ml seawater, that would already be 10 million viruses in that 1 ml of seawater. So these are, just to give you an idea, these are nanostructures all around us. And if we had all the viruses in the world, it would be more than the number of stars in the universe. If we had all these viruses in the world lined up end to end, it would reach 200 million light years into space. So they are all around us. And the other thing is that they have not just been around us, they have also helped shape human evolution. So we are what we are today because of several viruses and other pathogens that have interacted with humans since the beginning of the human race. There are a lot of genes in the human body that have come from viruses that have infected us over the years. It is hard to believe that right now, but a lot of genes, like the one that encodes for placenta, which is the sac that babies are developed inside the body of the mother, that sac, the gene encoding that sac, comes from a virus. And originally that gene was designed by nature to help a virus fuse to a host cell. But then evolution took place and this particular gene evolved to help the baby fuse to the mother's body. So it's hard to think of our existence without existence of viruses and our interactions with them over the years. I told you they're all around us, but also they are around us in different shapes and sizes. This is just a cartoon to give you an example of how how many different shapes of viruses exist. We have spherical ones, rod-shaped, complex, polyhedral, and each comes with its own property. So two that you have heard of a lot recently is the coronavirus, the novel coronavirus 19, which is a spherical virus, and Ebola, which is spindle-like. So two very different shapes and sizes, but both lead to infectious diseases, which makes it imperative to understand what these viruses are. It could be as small as the common cold that makes you sick for a day or two and then goes away, to something as dangerous as the Ebola virus, which can even lead to death in 34% of the cases. So it's important to understand that this is quite a complex biological entity, the virus, and it has several layers to it. It infects all kingdoms of life. So here's an example of a young girl infected with the poliovirus. That's a human virus, of course. We also have viruses that infect animals, such as dogs, the rabies virus. Plants are also infected with viruses from time to time. So when you see a plant which has leaves that are wilted and show light coloring, that's a plant infected with a virus. Like in this case, it's a tomato plant infected with a virus called tobacco mosaic virus. And interestingly, viruses infect other germs as well. So viruses infect bacteria and other viruses. In our body, we have a microbiome, which keeps our digestive system working. So in our microbiome, we have bacteria and we have viruses. And a lot of times they infect each other to keep our microbiome in check. So it's a very complicated system and we need to understand it. We need to learn from it. and see what we can apply to nanotechnology. So talking about understanding viruses, the first thing to note is that they are in the nanometer size range. Here are a few examples of names that you might have heard of: the poliovirus, hepatitis B virus, and they're arranged in the order of their size. So we have something that's 30 nanometers in diameter versus something that's like a really long rod, or almost a micron in length, the bacteriophage that's drawn over here. Nanometer, you know already what it is, but just to give some context, a nanometer is 1,000 times smaller than the width of a strand of hair. So it's really, really minute and not visible to the naked eye. Another property of viruses is that they are very regular and symmetric. Now, what do I mean by symmetry? It's almost like a football where you have these hexagons and pentagons that are arranged in a symmetric fashion, giving rise to the football. So similarly, viruses, and especially spherical viruses like the coronavirus model shown here, are repetitive units arranged in a very ordered fashion. Why it's important is that when it comes to design in nanomedicine, in nanotechnology, having one building block which can be repeated several times over gives you a high level of control, a high degree of control to a structure that you can make. So it's really just one building block that has to be arranged in a specific kind of symmetry. And this is why viruses are quite easy. It's quite easy for a virus to infect a host cell because it's actually just making one or a few proteins that are repeated several times over to make a virus capsid or a virus shell. Another thing is their self-sufficiency. So like I said, if you have one building block, in a lot of cases, for a lot of viruses, the building blocks would spontaneously assemble to make a capsid. So what you see in the simulation is one example where this blue colored building block is coming together just by Brownian motion, just by being in the same test tube, making a shell, which is a spherical shell, because that is the most stable form for that code protein, for that building block. So this self-assembly gives another property to the virus, making it quite easy for it to infect and makes copies of itself and propagate. They're really stable. Most viruses survive in harsh environments, which means they have to be very stable to survive outside. But at the same time, when they infect, they are malleable. They break apart, they release their DNA or RNA, whatever nucleic acid content they have, and lead to an infection. They're biocompatible, which means that they are not toxic like a lot of other nano materials. They are biological at the end of the day. So you know already for coronavirus, using soap and water is enough to destroy the virus completely. So at the same time as being stable and tough, they're also quite easy to destroy. And these are the sort of properties that make them attractive in the field of nanotechnology. We of course, have to keep learning about viruses, learn about ways to fight viruses because they cause infectious diseases. But at the same time, learning about some properties of viruses that can be applied in nanotechnology is also important. So I talked about infectious viruses. Now I'm going to give you two examples where you'll see that viruses can actually even be therapeutic. One is oncolytic virus therapy. So this is used for treatment of tumor cells, where viruses are used like a nano killer, a nano machine, where it's asked to infect and kill tumor cells. In cancer, what you really want is killing of tumor cells. And viruses are really good at doing that. So a therapy called Immunologic is available, and it's very recent. It was approved in 2015. So it's at this time, the only oncolytic virus therapy but it is one that uses viruses to benefit or for the advantage of mankind. Another one is virus-mediated gene therapy. So what is gene therapy? Gene therapy is when you have to deliver a gene to a damaged cell to make it healthy. And this example here talks about an inherited disorder called, it's a rare inherited disorder of the eye where the patient loses vision. So by using gene therapy, by supplying a healthy gene, the patient's vision is restored. And in this case, the healthy gene is supplied by a virus, because to think of it, a virus is nanometer in size, able to get into cells, able to deliver whatever is inside it, and biodegrade. So it is a good sort of container to deliver a gene to its target location. Again, it's a very recent approval. It's a therapy that's right now only approved for a very few diseases. And it's a very heavily regulated field. And one of the reasons why we regulate this field heavily is that these viruses, at the end of the day, are replicating, they're infectious, and there's so much damage that can be done by working with an infectious virus. Here's an article that came out earlier this year that points out that there are concerns about using viruses for gene therapy, because in this study, it was a very controlled animal study. In this animal study, they found out that there were some adverse effects of using viruses for gene therapy. And that is why the field really needs to be regulated. But now I'm going to introduce to you a concept that takes away the challenge with using viruses. What if we were not working with viruses, but particles that are virus-like, non-infectious, but having all the good properties, let's say, of the virus? And that's what the rest of the talk is going to be about, virus-like particles. Now, when we say virus-like, it's almost like having a container without its content, or having a snake without venom. So you have removed the negative parts of this entity and have kept only desirable properties. On the outside, the structure is very similar to the virus, but on the inside, it does not have any nucleic acid, any DNA or RNA, which is what leads to infectivity. So in this case, a virus-like particle will be something that's stable, it's robust, it leads to an effective immune response. So that's something I'll come to later, but it's something that leads to a good response from the body, like a vaccine, but does not have any potential to be infectious. How do we make these? A very quick, just one slide, a very quick overview of how these virus-like particles are made. In my case, we work with plants, but it can be any expression system. So you can use any expression system available globally. The first step is what we call molecular cloning. So that's to express genes. And these genes encode for viral proteins.
Pooja is not audible. Is there any problem? Brenda?
Oh, sorry. I thought it was fine.
I can very well hear. I don't think
Okay, now we can hear you. Okay, just go ahead. Thank you.
Okay. So going back to the how do we make these virus-like particles? So in this case, it's a three-step process. The first one is molecular cloning, which means that we design the genes that encode viral code proteins. So what we want is just the shell of the virus, not the interior. And to get the shell of the virus, we need the building blocks. The first step is to design genes that encode those building blocks. The second step is to express those genes. And in this case, it's expressed in plants. And the third step is to purify those particles. And what you see here is an electron microscope image of these particles. It is quite a well established process, and is being done in a number of places around the world. So what you end up with are virus-like particles, or VLPs, that are not infectious, easy to produce, and biodegradable. These type of virus-like particles or vaccines or VLPs are used for vaccines. And I've put just two examples of commercial products, two vaccines that are very well out there, they're in the market. What they do is that instead of using an infectious virus, they use the virus shell. So on the outside, the shell is identical to a virus. When this is administered as a vaccine, antibodies are raised, the immune system responds to it, and that leads to immunity against the particular target virus. So it's a method that's being exploited a lot to generate vaccines. And maybe you all saw this picture before. It was featured in all the leading newspapers recently to illustrate the different techniques that are being used to generate coronavirus vaccines. One of the techniques is this VLP technology, where what people are trying to do is make the coronavirus VLP. So it would be on the outside identical to the coronavirus, but on the inside it would be non-infectious or empty. As a result, if this vaccine is injected, a person's immune system would develop antibodies and immunity to coronavirus without ever having encountered coronavirus. So a VLP is a technology that is definitely used for vaccines, and we see that today. But what we don't see on the market yet, but has a lot of potential to come in the years to come, is are the applications that I'm going to share now. So these virus-like particles are being developed for a number of applications in material sciences. And I'm going to talk through some of these. The first one is an electronic nanosensor. So what is being done in the field these days is that the VLP, or virus-like particle, is being used as a scaffold. On the surface of this virus-like particle, very controlled mineralization can be performed. So gold nanoparticles, silver nanoparticles, silica, and other metals can be arranged on the VLP in an ordered fashion. This is a property that's desirable for material sciences, for nanotechnology. And one of the applications is the design of nanosensors, because having ordered nanoparticles of gold leads to conductivity, properties of electrical conductivity, which is otherwise hard to achieve because you don't get this kind of ordered display using any other synthetic method. So to reiterate, we have precise control on design and a very high degree of tunability using a VLP scaffold. Another application is that of a nano reactor. So think of a VLP as a little container that's nanometer size, that's a nanometer in its dimensions. So it gives you a very high degree of control over any catalytic reaction. You can almost control what ions are being put inside this nano container, leading to whatever catalytic reaction. So here is an example where very specific conditions needed to be achieved for generation of iron oxide from iron ions. And this specific condition was only able to be achieved by using a nanoreactor such as a VLP. On the right is another example where two conditions were used to encapsulate or to package different proportions of molecular cages. For simplicity, let's just say these are small blocks that needed to be packaged. And by just tuning the conditions used, the proportion of what's packaged inside the nano container could be modulated. So for material science, again, to have this kind of control and tunability is something that's desired and not achieved in any other way. And finally, two-dimensional and three-dimensional arrays. So again, another material science domain is the synthesis of arrays that would have properties that can be applied in an ordered fashion. VLPs are able to do that. VLPs are able to act as Legos or building blocks that can be arranged in an ordered fashion to give desirable properties. For simplicity again, let's just say this other molecule in green is something like glue. So by having the right glue and the VLP, you're able to get an assembly of VLPs just in the order that's desired for material science. So again, for VLPs, having the high degree of stability and the high degree of control and tunability is what makes them desirable. Shifting gears to nanomedicine. Now I'm going to take you through a few examples of how these VLPs are being used in the field of nanomedicine. One application is immunotherapy, and that is a little bit related to the use of VLPs as vaccines, what I mentioned earlier. So in immunotherapy, what's desired is a response from the immune system of the patient. Immune system is already primed. It has all the tools to attack cells, just that it needs some priming from time to time. So what VLPs are used for is presentation of antigens that can prime the immune system. These antigens alone are not able to lead to the desired effect. But when the antigen is presented on a VLP or presented on a cage, on something that's repetitive and something that's in the nanometer size range, it leads to the desired immune response. It's a bit like the Battle of Longaville, Rajasthan, that we read about in history. In 1971, when the Battle of Longaville was held, the Indian Army had 120 soldiers, as opposed to over 2,000 soldiers on the other side. Just as far as the numbers are concerned, we had no chance. But by cleverly arranging the 120 soldiers in formations, by using different approaches like the Air Force, like land, like different gears that were available at the time, the desired effect was reached, and the outcome was that this small 120 soldier army was able to come out as the winner. So that's kind of what we are doing in immunotherapy. We are trying to put this antigen in an arrangement which enhances its value, which enhances its effect, and leads to a response from the immune system. And VLPs are what allow us to add value. Another example is gene therapy. So what happens in gene therapy is that we need to use genes to replace a missing or a malfunctioning gene in the patient. It is required to deliver the correct copy or the healthy copy of the gene to a targeted location within the patient's body. So for this selective targeting and for protection of the gene until its delivery, VLPs are ideal. It's almost like delivering a letter. that needs to be sent to a very specific address and protected all the way. VLPs are ideal because, as we saw, VLPs are nanometer size range. They're able to get inside the body, get to a location, very stable on the outside, very malleable on the inside. So all the properties of VLPs make it desirable as a vehicle for gene therapy. A number of adenovirus vectors are being used in clinical trials. So it's really research that I'm bringing to you that's very, very recent and is going through a lot of research and development to get to commercialization and get to becoming viable products. So maybe in a few years from now, you will hear more of these words, which are still a little bit at the research stage right now. Drug delivery. Here's another example. So for a lot of applications like diagnostic imaging or MRI, one needs to provide a contrast agent to the targeted location. Or say in chemotherapy, the chemotherapeutic drug has to reach the targeted location. A lot of times patients have side effects because the drug goes everywhere in the body, even damaging healthy cells. That's why cancer patients that undergo chemotherapy lose their hair and lose any other rapidly dividing cells, like their immune system becomes weak. If there was a way of targeting the drug to one location and not affecting anywhere else in the body, it would minimize a lot of side effects and toxicity. And that's what VLPs are being designed to do. So the rationale is that the VLP can be filled with the drug or the diagnostic agent because there's space on the inside. And the surface can be tagged with proteins that would specifically take the VLP to the target location. So by selective tagging, we are able to ensure specific delivery of a drug or an imaging agent, thereby minimizing side effects, minimizing toxicity, leading to the desired drug delivery outcome. And another one is called magnetic hyperthermia. So this is an experimental treatment for cancer that works by using magnetic field to kill tumor cells. What it does is that it applies magnetic nanoparticles to the tumor, applies an alternating magnetic field from the outside. The magnetic nanoparticles heat up and destroy cells around it. So in principle, it is a good technology, but the challenge still is of delivering the magnetic nanoparticles to the target location. And that's where VLPs have an advantage. So what can be done in this experimental therapy is load the VLP with the magnetic nanoparticle, send it to the target location, apply magnetic field from the outside, and lead to killing of tumor cells. Again, this way, side effects are kept to a minimum, toxicity is kept to a minimum, and selective targeting is achieved. This is also an experimental therapy at this stage, very much in the research labs and hospitals in countries like Germany. But again, it needs more development, but it has a lot of potential of becoming a valid modality for treatment of cancer for a lot of tumors. So that was a lot of information that I want to wrap up. I tried to make it quite high level, but I included references on each slide if anyone's interested to read more about it. In general, the message that I want to deliver is that we have to understand viruses. It's the first step in turning our enemies into our friends. We have to understand about viruses to be able to develop vaccines, but also to be able to develop nanomachines for uses in nanotechnology. Virus-like particles are great for applications in material sciences and in nanomedicine, and they work by modifying at 3 levels: the inside of the particle, the interface of the particle, and the outside of the particle. And this level of precision is now possible in the field of nanotechnology. It wasn't the case a few years ago. And finally, I want to reiterate that virus-like particles bring a number of advantages for uses in nanotechnology, and specifically plant virus-like particles, which I talked about today, have advantages that make them almost ideal for uses in nanotechnology. With that, I want to wrap up and thank the places that I've spent my last 10 years working at. And I've learned a lot about this field from these places. John Innes Center in England. I worked with Professor George Lomonosov, who was my PhD advisor. He's one of the pioneers in this field, learned a lot from him, and still is the leader in the VLP field. Indiana University, USA was where I did my postdoc. There's a very strong virology group over there. And a lot of the material science applications I talked about come from labs at Indiana University. And finally, my company Medicago, where I do my day job developing vaccines for infectious disease. And we all know how important that is in a time like today. So I would like to thank these places where I spent my time. And just before I wrap up, I wanted to end on a slightly philanthropic note and introduce an initiative called Maskit India. It's an initiative started by two doctors in Noida. But what I want to bring to the group here is that we are at a time where people, especially the lower income strata of our society, need help, protection from coronavirus disease, and not everybody is able to afford masks. So Maskit India is developing, just using local resources to provide masks to the economically weak sections of society. These masks are free of cost, designed scientifically by providing several layers of the fabric so that people who don't have the knowledge of how a water droplet can be transmitted, they can still be safe. The reason I bring it up here is that it's an initiative that anybody at any level can get involved with. So if you feel like doing a little bit for the society, please reach out at the address here. It's something that you can get involved with while staying at home, using resources that are available locally to help people out in your local neighborhood. There are a number of Maskit centers around the country, and it's an initiative that's close to my heart. So I thought I'll just plug it in here. With that, I would like to end and welcome any questions. Thank you, everyone.
So any questions, please, from anyone? Or Vrinda, if there are any online question, you can, moderate them and ask if anybody.
Dr. Pooja, I would like to ask one question.
Yes.
I am Dr. Anuj, actually working in this Mechatronics Center, Center for Advanced Studies. So you would be happy to know that we are working a lot along with the state medical colleges to develop few products which can be helpful in sanitizing the various equipments and all those things. So I would like to ask one question. Do you have any study where we can found that this virus is being killed with the help of ultraviolet light, or so if it is being exposed to ultraviolet light? Generally, we are using here type C. It has wavelength from 100 nanometer to 280 nanometer. So it is helpful for killing this virus?
So this is something that I haven't come across. For coronavirus, all the studies that have been shown foolproof that you can destroy the virus is by using soap and water. because the viruses have a lipid layer, and you can only destroy the lipid layer with the soap and water mixture. Ultraviolet should, it could be one way of inactivating the virus. I have not personally come across a study like that, but I know that ultraviolet inactivation is used for making a lot of vaccines, where they take the virus, infectious virus, kill the infectivity by using ultraviolet radiation, and use the weak virus or the attenuated virus or the inactivated virus as the vaccine. So the rationale is there, but for coronavirus, I'm not sure. And I suppose you will be doing a lot of studies to confirm it.
Yeah, thank you so much.
Dr. Pooja, I have one question.
Okay, I have a question. And before this question, I would like to thank you a lot. You have given a good exposure to this area. Okay, my question comes from a scientific point, where you told the coronavirus size goes to the nano scale, around 60 to 100 nanometer. But in the visible range, when we see the wavelength of the light, it lies between 4 nanometer to 700 nanometers. So how could it be possible to see the nano size structure which is lesser than the visual range wavelength of the light? And second thing, if we can visualize it, is there any synthetic or something that we use with the virus to visualize it over the microscope?
That's a good question. So visualization is possible by electron microscopy, where the viruses are stained with uranyl acetate. This is a process called negative staining. So if a heavy metal is put and a beam of electron is put on the preparation, anything that's not blocked by the heavy metal is visualized. So we don't get resolution to 1 nanometer, but we get the rough structure. Like the image I showed of the virus-like particle, that was electron microscopy. For higher resolution than that, a technique called cryo-electron microscopy is used, where the viruses are frozen, and because of the freezing of the virus in a block of ice, the resolution, the electrons are able to go through the block of ice to be able to give better resolution. So you're right that there are limitations, and a regular light microscope will not do it. We have to go to electron microscopy, and that to cryo-electron microscopy for the best resolution for visualizing these nanometer particles.
Okay, thank you.
Dr. Pooja, I have one question. Like,
Yeah.
as you told that for VLPs incorporation, you use agrobacterium. So is it oriented to nuclear genome or chloroplast genome? Since both have a different property.
That's right. So we use a technique called transient agrobacterium infiltration, where actually it goes to the cytoplasm. So it's not nuclear or chloroplast. It's transient, which means it doesn't, it's not like the whole plant gets genetically modified. It's only that the leaf or whichever part of the plant gets agrobacterium, only that part will express.
Okay.
So it's a new technology again, and it's a very fast. Like for the other ones, you said that lead time is longer, but this one is a very fast way of getting expression of protein.
Okay. Thank you.
Okay, ma'am, I would like to narrate some questions from our students and attendees that I'm narrating it. That if someone is vaccinated against one virus, so is that person protected from other viruses too?
So the answer to that is no. It's very specific, and that's the reason why when infants are given a vaccine for polio, they also have to be given a vaccine for MMR or BCG. Every virus or bacteria is very specific, leads to a specific immune response, and you can't really have one vaccine that fits all. It has to be very specific for each disease. And it's also something that we are using to our advantage because we are working with virus-like particles that are not infectious to humans.
Okay, if we developing the vaccine, so it takes several months to develop a vaccine against a dangerous virus.
Yes.
So what if some new viruses emerged? Like, so it will take some other several months or year to develop a new vaccine for that virus.
So that's true, and you're right that it takes time to develop vaccines. One of the reasons is that you have to test the safety of the vaccine, the efficacy, find the right dose, monitor subjects for a long time. So development of vaccines takes time, but we can't stop after developing one vaccine. We have to keep researching so that we are ready for the next challenge. It's something in biology, we call it arms race, that it's like a race between the virus and the human, that we develop the vaccine and then there's another virus or another mutation. So there has to be it's almost like a race between the two. We have to keep on top of it. The more research we do, the easier it will be to develop a vaccine if a new virus emerges. So doing a lot of background research like it is being done in different parts of the world is absolutely necessary. I think we need a lot of investment in research and technology, and we need to keep working towards these challenges.
Yes, that's very true, ma'am. So personally, I enjoyed very much this lecture. As I'm a petroleum engineer, so many questions and so many confusions are in my mind due to this virus-like particle, that is nanoparticle. I'm taking it as a nanoparticle. So basically, I just take a course of MEMS technology, MEMS devices. So I was happy to listen that VLP can be used as a sensor, like modified version in sensors, nanosensor. So, and we can can we use these VLPs nanoparticles in petroleum? Is they having properties of cooling or lubrication and like this? So can we use in it like in fluid, like drilling fluid or cooling fluid or lubrication fluid that could not hamper the human health? So could it be?
Yes, I think so. I try to follow the field. There are so many labs around the world working on this. I came across a very recent work where a VLP was used like a bioreactor to create hydrogen. We know that there's an energy crisis and we need new sources of energy. So by using the VLP, they were able to generate hydrogen, which is a fuel that we need as a replacement to fossil fuels. So just like you're right, it is a nano container, it is a nanoparticle, and there's all sorts of cool chemistry that can be done around it. So for petroleum, I don't, right now I don't envisage how we would make the connection, but it's a field to follow.
No, we can yes, we can use these type of nanoparticles in developing some new type of nanofluids, so that the manpower who's working on the site cannot be affected.
Yeah, like with the hydrogen, it's a source of energy. It would lead to saving fuels.
Yes. So thank you, ma'am. This was a quite interaction. Thank you.
Well, it's my pleasure. It's the first time I'm talking my science to a person with petroleum engineering background. So I'm glad to hear your feedback.
Thank you, ma'am. Thank you so much. Thank you so much.
So are there more questions, Vrinda?
Yes, there are a few more questions which attendees have typed in Q&A chat. Let me read it to you. They are one-liners. Somebody wants to know what is the size of coronavirus?
120 nanometers. So I don't know that how that information will change anything. It's in the nanometer range. It's 120 nanometers.
And is it the case that coronavirus can pass from animals to humans, and is it possible?
There's conflicting evidence on that. So yes, people have published. It's a new field, so we have to trust what we read, what other people have discovered. So there have been papers, publications that have shown that coronaviruses could pass from cat to human, from dog to human, from even a big cat, a tiger, I think, to human. So it's things that need to be, like with any other scientific evidence, you have to check it, you have to repeat it enough number of times to be confident in the result. But so far the publications say that yes, the same virus can pass from animal to human.
And one last question is, can we use some kind of sound technology for coronavirus?
I'm not aware of that. I think the foolproof way would be to develop a vaccine.
Okay, one question which I want to know is, how much time does it take to actually develop a vaccine, and then how much time does it take to really reach to people? Will it be affordable? So all of these questions will come to mind.
This question will be answered in time. So in general, the process takes years, but because of the pandemic, a lot of the steps have been condensed, and all the bureaucracy has been reduced. Normally, to go through phase 1, phase 2, phase 3 clinical trial, then to the market, it takes few years. But now we are already hearing companies that will say that they'll be able to sell their vaccine in 12 months. I know 12 months seems like a long time, but for a pharmaceutical product that's so new, 12 months is a short time. You have to check that it's safe, and you have to have all the data before we start injecting people. And the other thing is providing the number of doses. So even if a vaccine is developed in 12 months, to have enough to be able to vaccinate everybody in the world is another challenge. So production, then it's a question of how fast can companies produce millions of doses.
Some of my student has actually dropped an WhatsApp message asking that if the impact of virus on any mentally ill patient is more than the normal.
Again, something I'm not aware of. So there have been studies that have said that if you are diabetic, if you are above 60, the impact is there. But on mental disability, I'm not aware of any specific adverse effect.
Okay, thank you, Pooja. Vrinda, I think for rest of the questions, you can send an email to Pooja, and Pooja can reply in the back because we are running really short of time now.
Okay.
Sir, one request from the student side, that can we get the PPTs? They are requesting. It's a very wonderful, informative session. So can we request Dr. Pooja to share her presentation to this?
I will do a PDF of like a simplified version and send it across.
Thank you so much. Sir, please.
Okay, so whenever there is such a nice or August gathering, director should not miss the opportunity to market his institution. So for those who are not belonging to Center for Advanced Studies, a brief information in couple of minutes that, we started this institution in 2017, and in a very short span of, about 2.5 years, we have already, made our footprints. The institute has set up a few very state-of-art laboratories, which are, very less in number in the country. We only operate in interdisciplinary, fields. So currently we are working in cybersecurity, machine learning, mechatronics, nanotechnology, and energy science and manufacturing technology. And our, all facilities. So we only offer postgraduate and doctoral programs. All the faculty are world class at our institution. And particularly related to this COVID response, the institute has really made a very innovative intervention by developing, multi-channel ventilator adapter, and we have worked in very close cooperation with Sanjay Gandhi Postgraduate Medical Research Institute at Lucknow. Also, we have come up with very low cost face shield, which is manufactured at our 3D printing laboratories. We have come up with number of sanitizing machines, both automatic and mechanical, which can be supplied at a very low cost. So these are our technological interventions, and I am very glad to inform that the design of all such products have been openly placed at our website, so that MSMEs or anybody who is interested in mass manufacturing for the use of common public can go ahead and do with it. So we are not taking any commercial advantage of all such developments. In nanotechnology also, we are following all WHO standards to produce the sanitizer. So the sanitizer is being locally produced by us at a very, very low cost, I tell you. So these are the things, and our research is world class. One of our professor is already working in in fact categorizing the corona patients based upon their X-ray images. So this is a machine learning kind of classification which he is trying to do, and this work is being done with Kota Medical College, KGMC, and number of other top medical colleges of the country. So thank you so much. Particularly, I wish to thank Pooja, who could take her time out, and particularly at this midnight hour, from Canada, to give such an informative talk, which can inculcate interest among our students, particularly nanotechnology students, because most of the time we work in technology. So now there is a dearth need to integrate the technology with biosciences as well. I also thank the panelist from St. Xavier Institute, Ranchi, Dr. Priya Srivastava, who also spared her time to be part of the panel and ask few interesting questions. I also thank Vrinda, Saurabh Mishra, and all other faculty members who were very keenly and patiently glued to their screen to hear you. So frankly speaking, it was a very wonderful experience, and I wish that in future also you will come up for further talks. I am receiving a WhatsApp message again from Dr. Priya Srivastava. She wants to very quickly ask one question. So I request Pooja to just satisfy her curiosity, and then we will wrap up. Thank you very much.
Dr. Pooja, I just want to, since the focus has been shifted to coronavirus right now, I just want to ask that if VLPs are being worked since long, what is the limitation? What are the limitations still for HIV vaccines till date?
HIV vaccine is a challenge because HIV mutates quite fast. So it's not like you have one target and you have a vaccine against the target. It keeps mutating, and it's a feature of the virus that makes it very hard to tackle. So we have a lot of antiviral therapies that can be used to treat HIV patients, but there is no vaccine out there that can prevent HIV, unfortunately. And like I said, we have to keep researching. So one day we will find a solution, but it is a complicated problem.
For sure.
I just forgot one thing, the Maskit India initiative that has been made by two doctors of Noida. One of the doctor happens to be father of Pooja. So I want to thank Doc Saab for such a wonderful initiative, and good luck to him. I want Pooja to convey our regards to him as well.
It's my pleasure. Thank you for your wishes.
Thank you.
Thank you.
So thank you. I think, are there more questions or should we wrap up?
I think we are running out of time, so I think we can wrap up now. We can take questions later, of course.
Okay.
Thank you. Questions will not end because this is such an interesting topic. Okay. Thank you very much.
That's why I keep on saying that, biology and technology should come together to make a difference.
Exactly.
Okay. Thank you.
Thank you very much, Dr. Pooja, for finding time. Thank you to all the panelists and attendees on Zoom, as well as who are watching live. Have a great day. It was a great start to the day with some interesting information on viruses. I hope you enjoy the rest of the day. See you then. Bye.
Thanks, everyone. Bye-bye.
Thank you. Thanks all.
I think we can good to close the session.